welcome. Thank you much, Winfried, for the kind introduction. I'm very excited to be able to present my work here today. And a short summary of what I'm going to talk to you about today, you can see already in this very first image here. This is, of course, an artist rendering in a way, what Jakob has been doing at the time. And what you can see here is in the blue greenish, these are localizations of molecules as they were diffusing along an axon of a cell. And you can very nicely see here this sort of ribbed structure, those sort of individual rings around this axon. And in the middle, there's a little bit cut out where you can see individual trajectories. And here also you can appreciate that there is like a periodic arrangement of these trajectories. And we believe that this is because the plasma membrane here is compartmentalized by submembrane actin cytoskeleton. Now, this is a little bit contradictory to our general model of how we understand the plasma membrane. And we all have in mind from the textbook, of course, the Singer-Nicolson model that after years and years of discoveries has led to the assumption that we have membrane proteins that are immersed in a two-dimensional liquid of lipids. And this membrane is, as such, fluid and continuous, which means that these molecules can move in all directions, basically in a random fashion. And that is exemplified here in a single molecule tracking movie of a single quantum dot coupled to a lipid as it moves around in a random manner by Brownian motion in a supported membrane bilayer. Now, quite quickly after the description of the membrane of fluid as continuous by Singer and Nicolson, first video microscopy, single particle tracking, and FRAP experiments found, much to the surprise, that indeed membranes exhibited in live cells much slower motion than you would expect from artificial membranes. And an example here is, of course, the famous Hop diffusion model from Aki Kusumi, who made very fast observations of membrane protein motion and found that they would be confined in smaller domains, and they would then jump from domain to domain over time. And it was rationalized that these domains here in purple and blue and green and yellow and red, they must be identical with what is seen in platinum replica M of the bottom plasma membrane and the associated cytoskeleton. You can see here the striated pattern here, that's cortical actin, that forms these little corrals. And Aki Kusumi believes that these are actually corresponding to these colored domains here on the left. But of course, so far this has been correlated work. Very convincingly, Kusumi also showed that if you attach a large bead to the cytoskeleton of a red blood cell, and you observe molecules just diffusing about, and then you then pull with an optical tweezer on that cortical actin, these molecules will continue to diffuse around, but they will move along, like they were caught in a meshwork. You can see that here. These are the diffusing molecules, and as the latex bead here is pulled, they are moving in that direction as well, but then at the new location continue random motion. Okay. This leads to the idea that there must be these corrals. Now, how is that rationalized? Well, you can of course imagine that you have a transmembrane molecule, and with its bottom portion inside of the cell, it would then bump into membrane-associated actin. So that only now and then it would be able to jump over that actin line with the so-called membrane skeleton fence model. On the other hand, there are molecules such as lipids, and these are also found to be confined. And that, of course, has no intracellular portion, so there's no way of interacting with the actin directly. However, we all know that there's many membrane molecules associated with actin. And if such a lipid would want to go from one of these compartments to the next one, they would have to sort of squeeze in between these pickets to find a new space to move around in. And that's the anchor protein picket model. These two models were put forward by Aki Kusumi and others to understand how membrane protein motion is compartmentalized in cells. However, of course, as we know, cortical actin is very dynamic, and all these observations necessarily had to be entirely correlated. So how do we go on about studying this? And we were very excited when in 2012, Zhao Wang and Ke Zhu found that along the axon initial segment, and indeed all along the axon, the cytoskeleton is organized in rings that are spaced by about 190 nanometer by spectral tetramers. So all along the axon, you basically have these rings every 200 nanometers that are connected by actin. And this is by now been described by many other laboratories and people, as you can see down here, and has been found basically from worm to humans in all kinds of neuronal cell types. And we were very excited to investigate and then also to see that if we look at actin in membrane protein motion, we could find that membrane proteins would be enriched in between these domains, right? So the localizations, of course, not the membrane proteins themselves. And we did so by doing high-speed, high-density single particle tracking, which means we just image a lot of single molecules as they move about, and then we can make maps like these, and we can measure diffusion coefficients with high statistical accuracy. How do we do it? And this is the heroic work by student David Albrecht, who is now a senior scientist at Max Planck. And what he did was he used neurons as they were just harvested from rat embryos, right? So then they can be seeded on gridded cover glass, as you can see here. So we knew where they were when we seeded them. We then transfected them with GPI-GFP and added probes to follow the motion of these membrane molecules, right? And we then, as the axon grows and the axon initial segment becomes assembled and these actin rings formed, over time, from day in vitro 3, over day in vitro 5, day in vitro 7, day in vitro 10, we always observed in the very same cell the axon as it assembles the cytoskeleton. Okay. This was an extremely difficult experiment, but it had beautiful results, as you can see here. So this is the axon after 3 days of growth. And you can see here, this is GPI-GFP staining, so the cell is transfected. Neurofacin as a marker of the early axon is not visible yet. And if we track the molecules, you can see they're all red. And here we have the diffusion coefficient from green for immobile to red for fast diffusion. You can see that all along the axon here, we have fast diffusing molecules. Here's the histogram. Now at DRV3, cytoskeleton starts to assemble. We see neurofacin, and here it's more yellowish. And then over the days, as the days progress in the very same cell, we have more and more green trajectories. And our histogram here moves to the left. And we can also look at the cumulative diffusion coefficient, and you can see that here where the actin ring is first assembled.